Hi, my name is Aaron Stewart. I'm an associate principal scientist at Lonza in Bend, Oregon. Today, I will give you a brief introduction to bioavailability enhancement, some key considerations for successful outcomes. And so the ongoing issue is just the sheer amount of compounds that are limited by solubility that ultimately leads to poor in vivo exposure. And so this is represented by the large share of compounds in the BCS class 2 and class 4 space, where solubility improvement is needed to achieve the desired exposure in vivo. And so many technologies are available to do this, starting with solid state alteration, whether that's a polymorph that has a higher solubility, amorphous systems such as amorphous solid dispersions, or even nanocrystals for size reduction, improved dissolution rate, and potentially solubility. We also can take a look at new crystalline compounds through solid form screening, which is now offered by Lonza. Taking a look at things like co-crystals and salt forms. Or you can also explore solvation techniques such as lipid-based formulations for improving bioavailability. At Lonza, we leverage an integrated approach to bioavailability enhancement, where we take a look at aspects of performance, stability, manufacturability, and drug loading. From a performance perspective, you need to be able to achieve the target in vivo exposure profile in vivo, leveraging things such as solubility enhancement, dissolution, and precipitation rates. From a stability perspective, your formulation needs to remain physically and chemically stable throughout the product shelf life. And from a manufacturability perspective, you need to be able to efficiently and cost effectively make your material without sacrificing performance or stability. And from a drug loading perspective, you need to consider the final drug loading in the dosage form drug product, such that you don't introduce complexities with respect to dosing unit size or pill burden. And so we can leverage both first principles approaches as well as historical knowledge for identifying which technology is best suited for our compound. Taking a look at the first principles approach, we leverage the compound's physicochemical properties to attempt to identify what the key barriers to absorption might be. So taking a look at things like what your dose, pKa, and biorelevant solubility values are, as well as estimating things like intestinal permeability, such that we can make calculations for what we think the rate limiting step to absorption might be. But it's also important to consider what you've done before, so historical knowledge based on what's worked and what hasn't, and distilling that into guidance maps based on drug physicochemical properties. And really the best approach is leveraging both of these in sort of an optimized fashion, such that you're taking into account some of the first principles approaches, because every problem statement is new, but also taking into account some historical data sets that you know worked for particular compounds and didn't for others. And so all of this really starts with identifying the problem statement, leveraging in vitro and silico tools, such that if you understand the key barriers to absorption, you might select a particular technology or formulate your compound in a particular way. So taking a look at things like basic property prediction and understanding the drug's physicochemical properties, any in vitro assessments that you need to do, such as solubility measurements or permeability assays, and then distilling that information down into some dimensionless number calculations such that you can attempt to bucket your compound into different rate limiting steps to absorption. And then if you have the information available, leveraging PBPK models such that you can identify key performance attributes or manage expectations or risks in vivo. So starting with the dimensionless number calculations. So these are calculations that you can make with routine data inputs that you would generate at the outset of a program. So solubility measurements of the crystalline or potentially the amorphous form if you're going to pursue an amorphous formulation, projected dose range and physiology or species, as well as estimations for permeability or biorelevant solubility. And you calculate things such as the dose number, the permeation number, and the dissolution number. The dose number is a measure of whether or not you are going to be solubility limited. So if this is a value greater than 1, your entire dose will not dissolve in a single GI fluid volume. The permeation number is a measure of how many times the drug can permeate over the course of GI transit, and so a measure of the permeation rate of the compound. And the dissolution number, likewise, is how many times can the drug dissolve to its saturated solubility over the course of GI transit. And these numbers are best utilized leveraging the fraction absorbed classification system, where they're interrelated into different rate determining steps to absorption. So starting with case 1, your dissolution rate limited, and so the dissolution rate of the compound truly governs absorption. In case 2, you're permeability limited, and so you're dissolving sufficiently fast and you're not solubility limited, such that you're just truly limited by the epithelium in vivo. In case 3, the solubility permeability limited case. This is by far the most popular case amongst BCS class 2 and class 4 compounds, where your dose number is far greater than 1 typically, indicating a solubility limit. And because you can only dissolve so much drug, that leads to a suppressed permeation rate. And so you have an interplay between solubility and permeability, which limits absorption. And so how do we take that initial information and in an attempt to identify the rate determining step to absorption and evaluate it in an in vitro setting? And so we have a fiber optic UV platform sort of portfolio of tests that we commonly employ for evaluating formulation performance based on what we think the rate determining step to absorption might be. And so maybe that's an amorphous solubility measurement. So you're evaluating the amorphous form of the drug. You then subject your formulation to standard dissolution testing, where you can get real-time data collection, taking a look at things like dissolution and precipitation. We have a dissolution permeation setup called the membrane flux test, where we can attempt to mimic passive absorption in vivo, which can be very useful if you can identify the rate determining step to flux in an in vitro setting and then relate that to an in vivo setting for a measure of biorelevance. And then we have more dynamic dissolution setups called the controlled transfer dissolution test, where you're metering drug out of a stomach compartment into a duodenum compartment at a biorelevant emptying rate, and taking a look at performance attributes such as dissolution and precipitation. And so how do we take all that initial information, starting with the pre-formulation calculations and the in vitro dissolution data of our formulations, and leverage it in a PBPK model to come up with outputs that ultimately lead to an optimized product concept? And so it starts with your in vitro dissolution data, any existing in vivo PK that you have, as well as drug properties. And at Lonza, the goal here is really to identify that rate limiting step to absorption. So starting with what is that physiological barrier to absorption in an in silico model? How is that tied to the critical performance attributes of your formulation? What initial product concepts would you propose to overcome that physiological barrier to absorption? And since we're often operating in a preclinical setting, understanding species comparisons as we go from animal study to animal study. And this ultimately leads to more informed formulation decisions, improved in vitro test design, retroactive in vivo data understanding if you have PK data to date. And ultimately, this leads to an optimized product concept through refinement and iteration. So at Lonza, absorption modeling is really a hypothesis-driven exercise, with the outcome being assessed risk and managed expectations. And so it's really important to identify that initial problem statement, because it can really lead to the optimized technology for your program, where you're able to assume some level of risk and realize the performance benefits that you need, but not too much, such that you're sort of overkill with respect to performance while assuming a much higher level of risk. And so it's important to understand that problem statement, because some technologies are better suited than others for overcoming certain physiological barriers to absorption. And so to conclude this short presentation today, it's important to recognize that BCS class 2 and 4 compounds continue to dominate the drug development pipeline. But good news, we have enabling technologies that are specifically designed to accommodate these types of compounds across a diverse physicochemical property space. And it's important to leverage the in silico tools, in vitro tools, and historical experience that you have to focus on the right technology with the highest possibility of success in the clinic. And with that, I'd like to acknowledge folks that have contributed to the work that I've described here today, and thank you for attending my presentation.